Imagine you're a forensic investigator walking into a crime scene. Or, hey, maybe you're a medical researcher looking at a patient's sample that contains just a tiny handful of viral particles. You have this microscopic amount of DNA, basically nothing to work with. How is it physically possible to analyze that? Direct analysis is completely out of the question. You can't sequence it, you can't test it. So, how do scientists actually solve this high-stakes biological mystery?
Well, welcome to today's Explainer. We're going to break down one of the absolutely most important techniques in molecular biology and biotechnology. The solution to that microscopic DNA problem, it was actually developed back in 1983 by a scientist named Kary Mullis, and it's called the polymerase chain reaction, or as you probably know it, PCR. And let me tell you, it is an absolute game changer.
So here's our roadmap for today. First, the tiny DNA problem. Then, the molecular photocopier. We'll look at the PCR recipe, walk through the three-step cycle, and finally wrap up with PCR in the real world. All right, let's dive right into Section 1, the tiny DNA problem. You know, before 1983, analyzing DNA was incredibly tough. It required massive, physically large quantities of a sample. If you didn't have enough, you were just out of luck. You couldn't run the tests. It was this huge roadblock for biology. But post-PCR, everything literally changes. We can now take extremely small amounts of DNA and amplify it into millions of copies. And we can do it in just a few hours.
Which brings us to Section 2, the molecular photocopier.
So how exactly do we go from a single tiny drop of genetic material to millions of copies? Well, the trick is mimicking the natural process of DNA replication that already happens inside living cells, but we do it in a test tube under super-controlled conditions. Dr. Mukesh Pimpliskar perfectly calls PCR a molecular photocopying machine, and I absolutely love that metaphor, because it's not just randomly growing DNA. It takes a very specific target segment, maybe from a plant, a virus, a human, and it amplifies it. It makes exact duplicate copies over and over again. It's literally just like putting a document on a scanner glass and hitting copy. And the sheer scale of the math here is just awe-inspiring. This amplification happens exponentially. So after one cycle of PCR, your single DNA molecule becomes 2. After 2 cycles, you've got 4, then 8. By the time you hit cycle 10, you have over 1,000 molecules. And if you run it for 30 cycles, you're generating over 1 billion copies from that original microscopic sample. 1 billion. Okay, let's look at how we actually make this happen in Section 3, the PCR recipe. All of this molecular photocopying happens inside this really cool programmable machine called a thermal cycler. But of course, you have to feed it the right biological mixture. First up, your template DNA. That's your starting sample with the target sequence. Next, you need primers, specifically a forward and a reverse primer. These are basically short synthetic DNA fragments that act as a starting point. Then we have the DNTPs. Think of these as the loose bricks or the building blocks used to build the new DNA strands. Finally, you need a PCR buffer to keep the chemical environment just right, and magnesium ions, which act as essential little cofactors to make the reaction work. But wait, I skipped one intentionally on that list: Taq DNA polymerase. This is the magic ingredient right here. PCR requires these wild, extreme temperature swings, which would honestly destroy standard biological enzymes. But Taq polymerase is special. It's a heat-stable enzyme isolated from a bacterium called Thermus aquaticus. And get this, where does this bacterium live? In boiling hot springs. Because of its natural environment, this enzyme can survive the intense heat of the PCR cycles without breaking down. It is without a doubt the engine of the entire operation.
So how does the machine use this recipe? Let's get into Section 4, the 3-step cycle.
Now that we've got our recipe mixed up in a test tube, we drop it into the thermal cycler. This machine runs a continuous repeating cycle of heating and cooling. Step 1 is denaturation. This is intense heat, about 94 to 95 degrees Celsius, which literally forces the double-stranded DNA to separate into 2 single strands. Step 2 is annealing. The machine rapidly cools down to somewhere between 50 and 65 degrees. This cooler temp allows our forward and reverse primers to bind specifically to their target sequences. And then step 3, extension. We heat it back up to 72 degrees. That is the absolute perfect environment for Taq polymerase to grab those building blocks and synthesize brand new DNA strands. One cycle finishes, our DNA has doubled, and then it just repeats the whole thing 25 to 35 times.
Now, just a quick aside, before and after those repeating cycles, there are some really important bookend stages. You start with an initial denaturation for about 2 to 5 minutes, just to guarantee the starting DNA is completely separated from the get-go. Then, at the very end, there's a final extension for 5 to 10 minutes to ensure all those billions of partially synthesized DNA strands are completely finished up. Finally, a hold stage drops the temp down to a chilly 4 degrees Celsius to safely preserve your sample until you're ready to take it out of the machine.
All right, moving on to our final part, Section 5, PCR in the real world. Okay, so you now have 1 billion copies of DNA in a tiny tube, but they're totally invisible to the naked eye. So to actually see your success, scientists use a technique called agarose gel electrophoresis. Basically, they put the PCR product into a special gel and apply an electric field. Because smaller DNA fragments actually move faster through the gel than larger ones, the DNA separates out. Once it's stained, the amplified DNA shows up as these really cool, visible, glowing bands. By comparing where those bands sit against a standardized DNA ladder, scientists can confirm the exact size of the fragment they've just amplified.
What's truly amazing is how adaptable this basic process has become. I mean, beyond conventional PCR, we have RT-PCR, which converts RNA into DNA first. That's incredibly important for detecting RNA viruses. Then there's real-time PCR or qPCR, which monitors the amplification live as it happens for super-sensitive quantitative analysis. We've got multiplex PCR, which amplifies multiple different targets at the same time. Nested PCR increases specificity, and digital PCR provides an absolute exact count of the DNA molecules. It's so incredibly versatile.
So why does all this matter? What's the big takeaway for us here? Well, the real-world applications of PCR touch almost every single aspect of modern life. In medical diagnostics, it detects viral and bacterial infections, genetic disorders, cancer mutations, you name it. In forensic science, it is the absolute backbone of criminal investigations, DNA fingerprinting, and paternity testing. Over in agriculture, they use it to analyze GMOs, catch plant pathogens, and improve crop yields. And in biotechnology research, it's fundamentally necessary for gene cloning and DNA sequencing. Now, as incredible as it is, PCR isn't magic, right? The advantages are super obvious. It's highly sensitive, very rapid, incredibly specific, and easily automated with just a tiny amount of starting DNA. But the limitations are very real too. Because it amplifies things so efficiently, it's highly susceptible to contamination. Literally, a stray skin cell from a lab tech can get amplified by mistake and give you a false positive result. Plus, it requires a lot of technical expertise to design those primers properly. And the thermal cyclers and reagents, they are not cheap. You really have to know exactly what you're doing. But ultimately, PCR remains one of the single most significant discoveries in modern biology. I mean, it entirely transformed biological sciences by allowing us to actually see and manipulate the microscopic building blocks of life. So I want to leave you with this thought. Since PCR has already revolutionized everything from forensics to disease diagnosis and acts as the absolute foundation of advanced molecular biology, what new medical and genetic frontiers will it help us unlock next? It's definitely a fascinating space to watch.